Okay, we have talked about amino acids, and we can remember that just listing those amino acids is a description of the primary structure of a protein. Now we're going to start talking about the secondary structure of a protein, which are the local domains. Now, in order to think about the secondary structure of a protein, we have to think about how these amino acids are coupled together. Okay, so here we've got two amino acids, right? This one is alanine, and this one is histidine. We're going to couple these two together into a dipeptide, right? A polymer of two amino acids. Now, the coupling process, the coupling reaction that occurs is a dehydration reaction. So we're going to lose water. And if you can remember that aspect of the chemistry, then you can get some clues into how these are going to actually couple together. So here we have the carboxy end, or the carboxylic acid end of our alanine amino acid. It's going to couple with the amine group from the histidine amino acid, and in that process it's going to lose water. So if we look at these two functional groups, we can take the OH away from the carboxylic acid and a hydrogen away from the amine group, and we're going to directly couple that nitrogen to the carbon. So when these join together, we think of this bond right here as being the really important new bond that's formed. This is the carbonyl carbon from the alanine, and this is the amine nitrogen from the histidine. And this bond right here is called the peptide bond, right? And it's what's joining the two amino acids. Okay, so we've got our peptide bond here. And that's the wonderful thing about coupling amino acids together. They are not coupled together by any sort of chemical reaction between the R groups. That wouldn't make sense, because then every chemical reaction between any different set of amino acids would have to be a different chemical reaction. We're taking just the standard part of each amino acid, the carboxylic acid and the amine group, and coupling them together. The R groups sort of sit out here to the side. Okay, now once we've coupled these together, we've sort of gotten rid of this carboxylic acid and got rid of this amine group. We've directly coupled them together and sort of destroyed those functional groups. And that allows us to describe sort of a directionality to this dipeptide. Now, since for any polypeptide, the peptide bonds are going to use up these functional groups, the start of the peptide and the end of the peptide are going to be the only places where we have any non-reacted carboxylic acids or amine groups. And we can see that with this dipeptide here, right? Here's an amine, here's a carboxylic acid, here's an amine, here's a carboxylic acid. But when I couple these two amino acids together, these two functional groups are lost and form a peptide bond. But we've got this loose amine group at the start and a loose carboxylic acid at the end. And so when we look at this dipeptide, we can say, well, here's the amine group, or the N terminus, and here's the C terminus, the carboxylic acid that hasn't reacted. And with proteins or peptides, that is the convention. The way we list amino acids, or the way we label out the sequence of a set of amino acids, is we always go from N terminus to C terminus. So this dipeptide here is an alanine histidine amino acid. It is not a histidine alanine amino acid. We always go from the N terminus to the C terminus, alanine to histidine. Okay. Now, once we couple two amino acids together, we can call it a peptide, right? Peptides we usually think of as short stretches of amino acids. Proteins we usually think of as longer stretches of amino acids, but there's no real designation for what, where that transition is from a peptide to a protein. Okay, so here's our N-terminus, here's our C-terminus, and this bond right here is the peptide bond. Now, if we start to think about secondary structure, the question we need to ask is, when we string together a whole set of amino acids into a peptide, are there any shapes that tend to form, three-dimensional shapes? And so we have to really go back to organic chemistry and start to think about what types of strain might be on a particular molecule as we rotate bonds. And when we think about the peptide bond, we think of it as a sort of a single bond here. And so for single bonds, we think they can freely rotate, and that allows for a lot of different possible conformations in a particular peptide. Now, what we're going to do to try to limit the problem that we have with this free rotation is we're going to think of the resonance structure that can form with this peptide bond. If we look, right, this nitrogen has a lone pair on it. That lone pair can come in, form a double bond between the carbon and the nitrogen, and two of the electrons from the carbonyl group can jump out onto the oxygen to form a negative charge. And so there is a resonance structure for the peptide bond that causes this carbon-nitrogen bond to have some double bonded character to it. And we know that double bonds can't freely rotate. So the way we're going to simplify our problem is to say, well, there's a bunch of different possible conformations for any polypeptide. But for this peptide bond, we're going to sort of freeze it in place. We're going to think of it as having enough double bonded character so that we don't have to worry about rotating that peptide bond. We just have to worry about rotating all these other bonds to think of all the possible conformations that might occur. So we think of the peptide bond as having double bonded character, and we can think of this part of our peptide as being sort of frozen in place as a plane. We'll show some examples of this in class. Now, if our peptide bond is frozen in place as a double bond, establishing this plane, really there's only two major bonds that we have to worry about rotating. Okay? And we're going to name those bonds phi and psi, and they are the bonds that flank the alpha carbon. So remember, here's our peptide bond. It's frozen in place. Here's a bond between the alpha carbon and the nitrogen. And that is a bond that we're going to call phi. It can freely rotate since it's a single bond. Here's the other bond between alpha carbon and the carbonyl carbon. That's called psi, and it can freely rotate also. So when we think about any particular polypeptide, strings of amino acids, we're going to think of the peptide bond as being frozen, unable to rotate. However, we can rotate phi and psi, these bonds that flank the alpha carbon, and we can get a bunch of different possible conformations for a polypeptide. Now, some of those conformations are probably going to be low energy, stable conformations, like this one up top. We think of this as sort of a trans-looking conformation, and it's because these R groups are on opposite sides of the peptide chain. That's probably favorable, so there's less steric strain occurring. And this sort of trans conformation is because we've got a particular rotation of phi and psi that allows for those R groups to be on opposite sides. Now, here's a combination of phi and psi that's probably not very stable at all. We've got more of a cis conformation with the R groups pointing at each other. If these R groups are bulky at all, they're going to be bumping into each other and causing a lot of steric strain. And so these are two examples of phi and psi bond rotations, one that looks pretty favorable, one that looks pretty unfavorable. Now, using this sort of strategy by freezing this peptide bond and looking at all the different rotations of phi and psi, you could imagine throwing that into a computer, right? And asking the computer, well, what combinations in phi and psi give me low energy conformations, and which combinations of phi and psi or bond rotations give me high energy conformations? And are there any patterns, any shapes that start to emerge as we look at really favorable bond rotations, right? Ones that have R groups sort of far away from each other to reduce steric strain and so forth. And so that's what we're going to do, right? We're going to ask a computer, we're going to feed it in a molecule and say, well, let's pretend the uh, um, peptide bonds are frozen in place because of their double bonded character. We're only going to worry about psi and phi, and we're going to ask the computer, like we did in organic chemistry lab with butane and cyclohexane, try out all these bond angles and show me some that are really low energy conformations. And, here's what we're trying to avoid. We're trying to avoid combinations of phi and psi that, create steric strain. Obviously, eclipsed conformations are bad. The staggered ones are going to be good. And what was found when this experiment was done, and we can sort of plot phi and psi against each other, right? We're going to look at all bond angles for phi and all bond angles for psi, and see which combinations of these bond angles give us low energy conformations. And what we find is if we plot this in what's called a Ramachandran diagram, we get these islands of stability, right? Here's one island of stability. The green and the blue indicate low energy conformations, and so we can look at, phi and psi around minus 90 here and maybe minus 60 gives low energy conformation, right? More of a staggered instead of eclipsed. And what we find is that when you look at a peptide that has these phi and psi angles, that it forms a helix, right? And this is where our alpha helix occurs under these bond angles. Now, this is one of the big islands. I call this helix island because there's a bunch of different types of helices that are present here, the alpha helix being the most common one. But here's another big island that we're going to talk about that has just very different phi and psi bond angles than the helices, and I'm going to call this sheet island, right? There's a bunch of different types of sheets that are present that form when you have these combinations of phi and psi, including a parallel beta sheet and an anti-parallel beta sheet. Now, if we look at all the different types of shapes that can occur through different combinations of phi and psi, we can start naming those different shapes. Alpha helix, parallel beta sheet, anti-parallel beta sheet. There's 310 helix, a right-handed beta helix, or a pi helix. There's sorts of ribbons and all sorts of different structures and shapes that form. And these are what we are going to call secondary structures, right? A string of amino acids that has particular phi and psi combinations that are energetically favorable tend to form these particular shapes, alpha helices, these different sheets. This doesn't mean the entire protein looks like a helix or a sheet. It's just a piece of it. A string of amino acids in the protein has a particular structure. Now, the ones that we're going to focus on are the first three, which are the most common secondary structures seen in proteins, the alpha helix, and then the two sheets, the parallel and the anti-parallel beta sheet. These have distinct phi and psi combinations, and they have distinct shapes that sort of solve some of the problems of energy that different conformations might encounter. And we'll see how that happens, first with the alpha helix and then with the sheets.